All right, welcome back. Lab 7 is all about the nervous system. So first, we'll look at the functional differences between the neurons and neuroglia. You'll be able to identify the important structures of a neuron, classify neurons, and understand how neurons transmit signals. There are two parts of the nervous system. We have our central nervous system and our peripheral nervous system. The central nervous system is the brain and our spinal cord, and the peripheral nervous system are our nerves, our sensory receptors, and our neuron cell bodies. And then two types of cells in the nervous system are our neurons. Neurons are the cells that transmit signals, and our neuroglia, which are supporting cells that help neurons function. Now we'll start going over the six types of neuroglia. First are astrocytes. These are the most abundant type of neuroglia in the nervous system. Astrocytes maintain the appropriate chemical environment to allow neurons to generate and transmit electric signals. Next, we have microglial cells. Microglial cells act very similar to immune cells. They are the resident macrophages in the central nervous system. They kill off any pathogens or invaders within the central nervous system, and they make up about 10 to 15% of the cells in the brain. Next, we have epidermal cells. They're also in the central nervous system in lined cavities that contain cerebrospinal fluid. These are ciliated supracuboidal cells that secrete and help move that cerebrospinal fluid. The last type of neuroglia that can be found in our central nervous system are the oligodendrocytes. These have processes that form myelin sheaths that wrap around the axon of neurons. Myelin is a form of fat that insulates the axon and helps speed up the transmission of electrical signals. And then this slide shows the two types of neuroglia in the peripheral nervous system. We have our satellite cells and our Schwann cells. Satellite cells are similar to astrocytes, and they help protect, cushion, and provide nutrients to cell bodies. And the Schwann cell are similar to the oligodendrocytes, and they produce the myelin sheath of the peripheral nerves. And there are many different types of neurons, and this image depicts a motor neuron. Motor neurons are characterized as multipolar. That means that they have several dendrites extending off of the cell body. Dendrites receive or receptor regions that pick up sensory information, and then sensory information is then processed by the cell body and sent through the axon hillock, and then down the axon in the form of electrical signal. The signal will then terminate at the axon terminal, which will either link to another neuron to continue that signal transduction or to another structure. This is a histology slide of a single neuron. You can see the main cell body and the dendrites coming off of it. And you can also see some of the neuroglial cells. The neurofibrils found within the cytoplasm of the cell provide support and helps transport substances within the neuron. And finally, we see our chromatophilic substances, and that is made up of a rough endoplasmic reticulum and is involved in metabolic activity of the neuron. So this image depicts a neural synapse. This is where two neurons will communicate and conduct the electrical signal. Electrical signals travel down the axon to the axon terminal, which will cause the release of a neurotransmitter from the presynaptic neuron, which will then bind to receptors on the postsynaptic neuron and allow for the electrical signal to continue to be carried. So neurons are classified based on the number of processes that extend from the cell body. We have unipolar or pseudo-unipolar, which means they have one process coming from the cell body. We have bipolar, which means there are two processes coming from the cell body. or multipolar, which means there are more than two processes coming from the cell body. So here we can see our multipolar, bipolar, and unipolar um, neurons. Multipolar neurons have several dendrites and one axon. Most of these are going to be motor neurons, and they're going to be carrying an impulse away from your central nervous system. Bipolar neurons have one dendrite and one axon coming from the cell body. These are rare. They're pretty rare, and you usually find them in the eye, the ear, and in your olfactory or your sense of smell. And these are mainly sensory neurons. And then lastly, we have our unipolar neurons. These are found in they have a so unipolar neurons have a peripheral process and a central process. The only distal portion of the peripheral process has receptive endings. And then this slide gives more examples of each type of structural neuron. So we see here our multipolar ones have lots of dendrites coming off the cell body. Our bipolar one has two um, processes coming off the axon and then the dendrites. And then we have our unipolar, which has our central process and our peripheral process, both extending off of one process coming from the cell body. And then neurons are also classified based on their function as well as their structure. So sensory neurons or afferent neurons bring signals to the central nervous system. And then we have our interneurons, which process these signals within the central nervous system. And then lastly, we have our motor neurons or efferent neurons, and they bring signals away from the central nervous system to the rest of the body. This slide shows the process of signal transmission in a reflex arc. The easiest way to explain this is with an example. So think of when you touch a hot stove with your hand. So your sensory neurons in your hand will pick up the sensation of heat from the stove. That sensation is picked up by the receptive endings of the peripheral processes of unipolar sensory neurons. And then that signal will travel up the axon via an afferent transmission to the central process of the axon, which is a synapse with the interneuron. That signal was then processed by the interneuron in the spinal cord. The interneuron will then decide what response is needed and will send a signal down the efferent motor neuron to the effector, in this case, the muscles in your arm and your hand. And that signal will cause a reaction and allow you to quickly move your hand away from the stove. All right, so next we'll tackle the brain and all its related structures. We'll be able to identify structures of the cerebral hemispheres, the diencephalon, the brainstem, and the cerebellum. We'll describe the meninges that cover the brain, identify the cranial nerves and their function, and identify differences between the human and sheep brain. So the brain has four main regions. There's the cerebrum, which is the visible outer portion of the brain divided into lobes. The diencephalon, which contains the hypothalamus, the thalamus, and the pineal gland. The brainstem, which has the midbrain, the pons, and the medulla oblongata. And lastly, we have the cerebellum, which means little brain, down here in the back. So the cerebrum has two halves. You have your left and right hemisphere, and it's also divided into six lobes. You have our frontal lobe, we have two parietal lobes, we have two temporal lobes, and we have our occipital lobe. And then the cerebrum also has raised ridges called gyrus or gyri, and shallow grooves called sulcus or sulci, and deep grooves, which are fissures. And then on the cerebrum, there are some special grooves. So we have our longitudinal fissure, which divides the cerebrum into left and right hemispheres. We have our central sulcus, which divides the frontal lobe from the parietal lobes. We have our lateral sulcus, which divides the temporal lobe from the frontal and parietal lobes. We have our parieto-occipital sulcus, which divides the temporal lobes from the occipital lobes. And then lastly, we have our transverse cerebral fissure, which divides the cerebrum from the cerebellum. So here's a representation of the grooves and the lobes found in the cerebrum. So up here in the front of the skull, we have our frontal lobe. Going towards the back, we see our precentral gyrus, our central sulcus, our postcentral gyrus, and then we have our parietal lobe here in the lighter in the this blue here. Keep going back, we see our parietal occipital sulcus here, between our parietal and occipital lobe. Then we have our temporal lobe, and then we have our cerebellum, and that's separated by this transverse cerebral fissure. We continue going down, you see the pons, the medulla oblongata, and the spinal cord. And then down here, we can see the difference between a sulcus and a fissure. A sulcus is a more shallow indentation, while a fissure is a deeper indentation. And then this image shows the top of the brain, and you can see again that longitudinal fissure separating it into left and right hemispheres. And then you can also see that central sulcus separating the anterior and posterior portions of the brain. And this is the same image as before, except it's a real life cadaver image of a human brain. Here you can see that really prominent longitudinal fissure down the middle. This slide represents the motor and sensory areas of the brain. So each function is separated onto different parts of the cerebrum. The motor area is responsible for movement. The prefrontal cortex is responsible for complex behaviors, personality expressions, decision making, and social behavior. The Wernicke's area is involved in processing speech, and Baraka's area is involved in the formation of speech. Vision is located in the back of the brain on the occipital lobe. Hearing and taste are on the lateral portions of the brain. And then lastly, our somatosensory region, or where we process sensations that occur anywhere in the body. So like pressure, pain, temperature, things like that. So next we have our diencephalon. The diencephalon has our thalamus, which relays information from the brainstem to the cerebrum. We have our hypothalamus, which controls the important body functions like sleep, thirst, hunger, body temperature, and coordinates pituitary gland function. And then lastly, we have our epithalamus, which is holds our pineal gland, which produces melatonin and helps regulate circadian rhythms and sleep. And then next we have our brainstem. So from top to bottom on the brainstem, we have our midbrain, which is involved in hearing, vision, temperature regulation, motor control, and our sleep wake cycles, also our circadian rhythm. Then we have our pons, which help to regulate breathing and relay signals from the medulla to the midbrain. And then lastly, we have our medulla or medulla oblongata, which controls autonomic functions like breathing, heart rate, and blood pressure, and things you just don't have control over yourself. And then next we come to our cerebellum. Cerebellum means little brain. It plays a major role in motor control, and it helps coordinate movement with proprioception. Proprioception is just your awareness of your body in space. So think of it as your place in space. This image is of the inside of the brain. So if you can see the fornix and corpus callosum are areas of tissue that separate the thalamus from the cerebrum. In between the fornix and corpus callosum is a cavity filled with cerebrospinal fluid called a ventricle. The anterior and posterior commissures are connection points that connect the left and right hemispheres of the brain. The corpora quadrigemina is in the midbrain is our reflex center involved in hearing and vision. The cerebral aqueduct is a region where the cerebrospinal fluid flows and then pulls into the fourth ventricle. The choroid plexus is a group of cells that produce cerebrospinal fluid. The arbor vitae of the cerebellum is the white matter of the cerebellum. Arbor vitae means tree of life. If you look at it here, it looks like a tree. The interthalamic adhesion connects the left and right portion of the thalamus together. The medullary body relays impulses to the thalamus. Next, we have our interventricular foramen, which connects the ventricles together. And then lastly, within the brainstem, you can see the pons, the medulla oblongata, and then lastly, the spinal cord. And then this is just a cadaver image of the previous slide. So again, you can see our ventricles, our cerebellum, that arbor vitae, the medullary body, the pons, medulla oblongata, and all the other things that were on the last slide. Then here is an image of the cerebellum that has been separated from the rest of the brain. In the middle of the cerebellum, we have the vermis, which is similar to the corpus callosum, and it joins the two halves of the cerebellum together. And then similar to the cerebrum, you also have lobes and fissures within the cerebellum. And then this is a cross section of the cerebellum, and you can really see the white and gray matter of the cerebellum. Gray matter is made up of neurons and their cell body, and the white matter is made up of the myelinated axons of the neurons. So next we have our meninges of the brain. There are three membranes that enclose and protect the brain. The outer layer is the dura mater, which means tough mother. It's the thickest and most protective layer, and it's made up of two layers within that. So there's the periosteal layer, which lines the skull bone, and then there's the meningeal layer, which is beneath that one. Next, we have our arachnoid mater, which is the spider mother. It's the middle layer that has a spider web-like extensions that extend into the subarachnoid space. And then lastly, we have our pia mater, which means tender mother. It's the thinnest layer that directly covers the surface of the brain. And here we have a visual representation of the layers of the meninges. Beneath the skull, we have the two layers of the dura mater, the periosteal and meningeal layer. Then we have our arachnoid mater, and then finally the deepest layer is our pia mater. So next we have our cerebrospinal fluid. So cerebrospinal fluid is produced by the epidermal cells in the choroid plexus of the brain. Cerebrospinal fluid acts as a cushion, and it's circulated throughout and around the brain, and it's held in our four ventricles. We have our two lateral ventricles. We have our third ventricle, which is located within the thalamus, and then our fourth ventricle is located behind the brainstem. Here is an anterior view of the brain. In blue, you can see the ventricles in the different places that the cerebrospinal fluid will flow through. You can see our two lateral ventricles, and below those are the third ventricle. Connecting the lateral ventricles to the third ventricle is the intraventricular foramen. And then in between the third and fourth ventricle is the cerebral aqueduct, which flows the cerebrospinal fluid from the third to the fourth ventricle. And then beneath the fourth ventricle is the central canal, which allows cerebrospinal fluid to flow down from the brain and into the spinal cord. Here is a lateral view of the ventricles. On the lateral ventricles, there are special regions called horns. We have our anterior horn, our posterior horn, and our inferior horn of the lateral ventricle. So now we're going to go over the cranial nerves. There's 12 of them. You need to know the name, their number, and their function. This is probably going to be the hardest slide for you guys to remember for this unit, in my opinion. The easiest way to remember them is to use the mnemonic. On occasion, our trusty truck acts funny. Very good vehicle anyhow. So that's what we use to remember the order of our cranial nerves. So here we can see the representation of our cranial nerves. There are 12 pairs, so one nerve for each side of the brain, but so 12 each side. So when we look at this, we start from the front of the brain, which is the top of the slide, to the back of the brain, which is the bottom of the slide. The first spinal cranial nerve is the olfactory nerve. Second is our optic nerve. Third is the ocular motor nerve. Fourth is the trochlear nerve. Fifth is the trigeminal nerve. Sixth is our abducens. Our seventh cranial nerve is our facial nerve. Eighth is the vestibulocochlear nerve. The ninth cranial nerve is the glossopharyngeal. 10 is the vagus nerve. 11 is the accessory nerve. And lastly, 12 is our hypoglossal nerve. So this shows the name, the number, and the function of our 12 cranial nerves. So olfactory is our first cranial nerve that has to do with our smell. Our optic nerve is our second one. It has to do with vision. Our ocular motor nerve is our third cranial nerve, and that lets you move your eye and adjust the size of your pupil. Next, we have our trochlear nerve, which is number 4, and that allows you to move the eye as well. Number 5 is our trigeminal nerve. That's the sensations of the face and helps with chewing. 6 is our abducens nerve, which lets you move the eye in abduction, so to the side. 7 is our facial nerve, which helps with facial expression in our salivary glands. The vestibulocochlear nerve is number 8. That has to do with hearing and balance. Our glossopharyngeal nerve, number 9, has to do with swallowing and taste. So think your tongue. Our vagus nerve, number 10, deals with our heart rate and our digestion. Accessory is number 11, and that helps you move your head. And then your hypoglossal is number 12, which moves your tongue. Here we have a sheep brain. It looks very similar to a human brain. Two key differences are obviously the size. It's much smaller than the human brain, but also the olfactory bulb or the olfactory nerve is much larger in the sheep than it is in the human. Here's another image of the sheep brain. The cerebrum or the cerebellum has been pulled away from the cerebrum. You can see that pineal gland that is responsible for our circadian rhythm. And you can also see the corpora quadrigemina, that is a reflex center for hearing and vision. Here's a medial view of the sheep brain with the left and right hemispheres separated from each other. It looks very similar to the human brain. A key structure here labeled is the cerebral funnicle. It connects the cerebrum to the brainstem, and it should know it is both present in human and sheep brains. And here's a cross section of the brain where you can see the ventricles and where the cerebrospinal fluid will flow through and throughout around the brain. So next, we'll start looking at the spinal cord. We'll be able to understand the structure of the spinal cord and then name the meninges of the spinal cord and state their function. So here we have a superior view of a vertebrae and the spinal cord going through the vertebral foramen. The spinal cord has the same meningeal layers as the brain. You have the pia mater, the arachnoid mater, and the dura mater. And then coming off of the spinal cord, we have our spinal nerve. And then within the spinal nerve is our dorsal root ganglion. The dorsal root ganglion is where the sensory neurons relay sensory information to the spinal cord. The spinal cord has 31 pairs of spinal nerves, one for each side. And then the spinal cord runs from your first cervical vertebrae, so C1, down to L1 or L2, so our lumbar vertebrae. And then it ends at the conus medullaris. This is a structure where the spinal cord ends. And that just means the end of spinal cord. The dura and arachnoid mater continue to run down to the sacrum past that conus medullaris. And then the pia mater ends at the conus medullaris, and then it'll turn into another structure called the filum terminale. And then lastly, on your spinal cord, we have our cauda equina, and it looks like a horse's tail, and it begins at the end of the spinal cord. So on the next few slides, we'll look at different sections of the spinal cord, starting at the cervical vertebrae, then the thoracic vertebrae, and then ending at the lumbar. Look at these a few times because you will need to be able to identify which portion of the spinal cord you're looking at for the practical. So this first one is an image of your cervical spinal cord and your cervical vertebrae. You can tell that it's the cervical vertebrae because the foramen magnum is present here. And then you can also see the medulla oblongata of the brainstem. And then this is a thoracic spinal cord. For this one, look for the dorsal root ganglion. They're going to be very big here, so that's a key way to know that this is our thoracic spinal cord. And then lastly, we have our lumbar spinal cord. Look for the cauda equina. It looks like a horse's tail. And then look for the filium terminale. And here we can see the gray and white matter of the spinal cord. Again, the white matter is the myelinated axons of the neurons, and the gray matter is the cell bodies of the neurons. You can also see the dorsal root ganglion and the ventral root. You can also see the pia mater, the arachnoid mater, and the dura mater to our meninges. And then the central canal is where the cerebrospinal fluid is going to flow down from the brain through the spinal cord. And then lastly, we have the dorsal median sulcus and the ventral median sulcus, which will separate the spinal cord into left and right sides. And then this is a histology slide of the spinal cord. The white matter is the darker purple, and then the gray matter is the lighter purple. And finally, we'll look at reflexes. So we'll explain the two types of reflexes, understand how a reflex will result in an action, explain the importance of reflexes, and then demonstrate how to test certain reflexes on individuals. So the two types of reflexes are somatic and autonomic. Somatic reflexes respond to sensations and causes your skeletal muscles to react. An example of this is when your doctor taps your knee and forces your leg to kick out. That's your patellar reflex. Autonomic reflexes are controlled by the autonomic nervous system, and they are involuntary. An example of this would be when you're really hungry and you smell food, your mouth starts to salivate, or when you walk outside, your pupils get smaller to adjust to the light. So these are things that you cannot control. Here's an example of a somatic reflex arc. You have a somatic stimulus. In this case, it's a nail in the skin. And then a receptor will send that signal down the sensory neuron to the interneuron. And then the interneuron will then send a response down the motor neuron and cause your muscles to move away from the stimulus. And here is an example of a monosynaptic reflex. A monosynaptic reflex does not have an interneuron. There's a direct connection between the sensory neuron and the motor neuron. And this is a polysynaptic reflex, so there are going to be multiple connections being made. A polysynaptic reflex does have an interneuron between the sensory neuron and the motor neuron. Then here's our last slide of the day. This shows a stretch reflex. This shows how antagonistic muscles are inhibited from acting to compose the intended reaction at the knee. So for this one, the sensory neuron is picking up the signal from the hammer tapping the patella and sending a signal back to the spinal cord. And then two signals will come back from the spinal cord. One will go straight to the quadricep muscles without going through an interneuron, and that will cause the quads to contract and extend the knee. And then the second signal will come through an interneuron and then down to the hamstring muscles, telling them to relax to allow the knee to extend.